Hi, I'm Dr. Danto, and today we'll be discussing a study about the effects of osteopathic manipulative treatment, or OMT, upon the brain and the possible benefits for OMT in Alzheimer's dementia. Alzheimer's dementia is what we call a degenerative disease of the brain. Degenerative diseases imply that we slowly break down in structure and function. Alzheimer's disease primarily affects our growing elderly population. As we can observe, the Alzheimer's dementia afflicted brain tissue is shrunken as compared to the relatively healthy brain tissue. This disease affects over 50 million people worldwide, and there is no known cure. A major player in Alzheimer's is the neurotransmitter acetylcholine. This nerve messenger chemical is affected, resulting in extensive loss of nerve cells and the formation of plaques, which are like scar tissue structures within the brain. There are also two types of proteins that play an important role in Alzheimer's, and they are the beta amyloid and the tau proteins. There develops a protein imbalance between them that reaches a tipping point, resulting in excessive accumulation of the tau proteins throughout the brain, which causes tangles between the nerve cells and the glial cells. These tangles interfere with the proper brain functioning. Over the years, scientists have developed two drug types for the treatment of Alzheimer's disease: acetylcholine esterase inhibitors, which decreases the breakdown of acetylcholine, and NMDA receptor antagonists, which affect the propensity for the amyloid plaque formation. I wish the treatment was long-term, but to paraphrase Dr. Anandakrishnan and his colleagues, these treatments are effective in managing the symptoms in the short term, but their longer-term treatment benefits are questionable. In a recently published article in the Journal of Osteopathic Medicine, Dr. Anandakrishnan and his colleagues reported a pilot study that describes how osteopathic cranial manipulative medicine, also called OCMM or cranial osteopathy, could be a potential adjunct treatment for Alzheimer's dementia. Cranial osteopathy has been around since almost as long as osteopathy itself. It was most significantly defined and explored by a DO named William Gardner Sutherland. As a medical student at the American School of Osteopathy in 1899, Dr. Sutherland was examining a skull that was disarticulated, meaning that the skull bones were dissected apart as pictured in the skull beneath his right hand. He noticed that two of the bones were beveled like the gills of a fish, and the thought struck him like a lightning bolt that this relationship indicated motion. He then set out to prove himself wrong, as good scientists try to do, and ended up defining osteopathic cranial manipulative medicine, which I will discuss deeper in a future recording. The Anandakrishnan and colleagues study was designed using rats as models. There were 3 rats in the treatment group and 3 rats in the control group. The treatment group received a special cranial osteopathy procedure performed under anesthesia called a CV4, which was repeated daily for 7 days in a row. The anesthesia was also administered to the control group each day. The rats were humanely euthanized after the 7-day period. I apologize to those that may be offended to find that animals are still used for scientific study, and I offer prayers of thanks and appreciation to the rats for their sacrifice. Finally, tissue samples were obtained from the prefrontal cortex of the rats, and the RNA was extracted from the tissues for sequencing. They observed multiple genetic expression changes in the treatment group. The 5 genes observed to have the largest changes were related to acetylcholine neurotransmission and most likely resulted in increased cholinergic neurotransmission. Dr. Anandakrishnan and colleagues proposed 2 reasons for the OCMM to affect the genetic expression. First, they thought that OCMM may improve the lymphatic fluid circulation. Lymphatic fluid is basically one of the ways the waste products of the body drain from the cells. Alternatively, They thought that the OCMM would cause a cellular stress that would change the RNA expression. I am not sure what they meant by stress, as the terminology that they're using is not usually used to describe OMT effects. Regardless, I believe that their theories are only partially correct, and I will explain why in what follows. You can be the ultimate judge of who you think is right, or if there's some combination that you can come up with, that would be great. Let's talk a bit about some relatively new insights that we've gained in neuroanatomy and neurophysiology. In 1955, Dr. Thomas Harvey took samples of Albert Einstein's brain. Despite the best efforts of many scientists, none had discerned any differences in Einstein's brain tissue that accounted for his fantastic intellect. Then, in the mid-1980s, Marion C. Diamond of UC Berkeley found that Einstein's brain had an unusual number of glial cells in regions responsible for higher thought. This was a major breakthrough. Previously, scientists thought glial cells functioned more like a skeleton and insulator for the brain. But we are now beginning to understand that glial cells have a much more significant role. It appears that glial cells can communicate with themselves and with nearby neurons. The glial cells also appear to have fine control of blood flow within the brain. It has been observed that when this function is lost by glial cells, there is a loss of the ability to form recent memories. When an osteopath like myself contacts a patient's head to perform cranial manipulation, we palpate through the skin and direct our senses through the underlying soft tissues right down to the bones. We perform motion testing on these tissues just like we would do anywhere in the patient's body. The big difference is that the examination has a much smaller range of motion. This range of motion has been studied and found to be reliably consistent with examiners and between examiners, and even instruments have been used and shown to reliably observe this cranial motion. What made me excited about the growing understanding of glial cell function and cranial osteopathy comes from neuroanatomy. When we manipulate the skull, there is an effect upon the head's soft tissues, and the attachment of these tissues has been mapped right to the glial cells. This means that performing cranial osteopathy on the outside of the skull most likely results in changes within the brain itself. According to the National Institute of Health, another significant component of Alzheimer's disease is that the glial cells fail in their duties to clear away waste, debris, and protein collections, including beta amyloid plaques. Based upon the understanding of the structure and function relationships that forms one of the foundations of osteopathy, I'd propose that cranial osteopathy improves glial cell function through myofascial continuity within the body mechanics. The improved glial cell function removes the waste products and debris that results in the amyloid plaque buildup. Additionally, the improved glial cell function supports the neurons to improve their function as found in the Nandakrishnan and colleagues' genetic expression study. For more information on aging and Alzheimer's disease, I will refer you to the NIH website. The information I've discussed is based on the scientific studies that I've reviewed, and there are probably others that have come along that might have different conclusions. Alzheimer's dementia is a subject matter near and dear to many of us for a lot of reasons. The plain fact of the matter is that as we age, our bodies tend to wear out. My job as an osteopath is to support the body to function at its best. Remember, our goal at Danto Osteopathic is to normalize, optimize, and thrive. Aloha.